Hi, everyone. I'm so happy to be here with you today. My name is Yevgenia, or I often go by Zhenya Korozorvitskiy, and I'm an associate professor of neurobiology at Northwestern University. And I am one of the scientific co-founders of a company called Neuroplastica. This is a synthetic biology-based platform company whose goal is to screen compounds that change your brain connections. So we're very early stage, and we're truly focused on identifying drugs that can help treat mental health and cognitive disorders, neuroplasticity, or activity-based remodeling of dendritic spines and synapses. We have a unique scientific platform that uses high-throughput bioluminescence screening to try to remove bottlenecks in very early drug prioritization. And this is all built around science from world-class leaders in thinking about neural remodeling and how the brain changes in development and throughout life. I've spent the last 25 years obsessed with studying neuroplasticity, starting at Princeton University, then at Harvard Medical School, and for the last 9 years or so here at Northwestern. We're a small and capital efficient company with programs with excellent partnership potential, spanning both small startups and large pharma. I don't need to spend a very long time reminding everyone that we're living in a pretty substantial mental and cognitive health crisis today, with a large fraction of our population affected, increases due to the COVID pandemic, first-line medications failing for a large fraction of patients, and for some conditions involving brain and mental health, no drugs approved at all. Looking a little bit more quantitatively, indeed, you can see that the unmet for central nervous system therapeutics, the unmet need for that is substantial from 80s and going on through today. And further, if you look at the drugs for CNS that are getting approved, you can see that for many conditions, they're accompanied by high rate of clinical failures. So why is that? A major problem is that we have quite weak models for complex brain health conditions. It's not very surprising. We're just not very good at modeling very much about the human brain. And because of that, we end up over-investing into drugs that will ultimately fail later. And so this leads one to consider the importance of a focus on phenotypic screening. If you know what it is you should be screening for. And to understand how we are approaching this, we got to do a little bit of a review of neuroscience and consider that everything that you live, everything you are, is processed inside your brain by billions of neurons that are wired together into complex structures whose complexity exceeds that of your computer and your iPhone, by literally trillions of tiny connections through which neurons communicate with one another. Many of these connections, called synapses, are contained in dynamic structures called dendritic spines. And these are really tiny. There's about 200 million of them in 1 grain of sand or so. And they are what allows us to think and feel and truly form the dynamic unit of neural computation. They're produced soon after birth in large quantities, refined in activity-dependent way, and then continue reshaping themselves in number and size throughout life. Given how fundamental they are, perhaps it's not surprising that many brain conditions, here I'm just using a few of them, are associated with various types of changes in the spines or their dynamics, oftentimes decreases. So can we then find drugs and find efficient way for identifying compounds that will modify specifically the dynamics of dendritic spines and this phenomenon of neuroplasticity? Seems like a really good idea, but it's been difficult, in part because plasticity is regulated by really complex biological pathways. Here I'm using a little cartoon schematic to highlight convergent pathways, actions in the nucleus that then feeds back to the membrane and reorganizes synapses and spines. So typical high throughput based approaches have by and large not been successful for this particular phenotype. Now, there are complementary high-throughput imaging-based approaches that have been attempted as well, and those two have been largely not very successful, in part because of relatively narrow dynamic range readout if you're focusing on counting dendritic spines, and also due to the enormous amount of data that you need at very high resolution in order to capture this. So we thought about this problem for a while, wanting to identify new compounds that regulate neuroplasticity, and ended up coming up with a very different idea of a bioluminescent sensor screen that senses neural activity, and when it reaches amounts sufficient to remodel the network, produces a bioluminescence readout that can be efficiently measured, that aggregates changes in dendritic spine density and the growth of individual synapses and spine. So how are we going to use that?
Yevgenia, we have some static every once in a while from you. I don't know what's in your background, but just wanted to flag it for you.
Okay, thank you very much. So how do we use it? We start with our biosensor, or actually it's now a relatively broad family of biosensors. And then we put it into neuronal cultures. And here I want to make a few asides. You can use mouse cell cultures, certainly human-derived pluripotent stem cells work. You could also use organoids, although we haven't done that yet. And we're currently validating the sensor use in vivo, which is actually quite appealing because you don't have the optical imaging problems with bioluminescence. And then the sensor expresses. We submit compounds or use existing libraries, and using a robotically assisted high throughput pipeline, relatively quickly generate large amounts of data that once we reach sufficient depth of the database, we hope to apply machine learning based algorithms in order to help us classify compounds with respect to effects on neuroplasticity. So the screen is relatively efficient and provides highly quantitative information that has a good dynamic range, and due to a little bit of baseline activity, even dynamic range in the negative regime, which has actually been turning out some very interesting biological results in our data. You can easily complement with various kinds of controls, including positive controls like the deep red line shown here, as well as various omission, sensor, and drug target controls. Just to give you a little bit more flavor, here's a small 4-plate screen that we have done. This was the first screen we did on a small FDA-approved compound library. And these are all 384 well plates that are about the size of your palm, right? So the screen miniaturized rather well. And you can see data then as z-scores, where each of these little squares represents a specific well with a specific number associated it for the amount of luminescence that was produced. And we were very pleased to see these data because not only were we able to identify multiple expected positive control hits, because this is a relatively well-studied compound library, but also we identified new ones and started exploring interesting negative regulators as well. And for this library, we also could identify the targets that these drugs act on rather well. And so this was kind of critical validation for us. And since this early screen, we've followed up with secondary screening of this library, as well as screening some other libraries, including natural products that have led to some very, very interesting observations, as well as starting to work with some proprietary compounds. I want to also point out here that we see this as a very early stage prioritization tool for compounds. And certainly in the context of the lab, we also bring to bear a lot of really useful follow-up mechanistic approaches, ranging from genetically targeted synbio-assisted proteomics we do, to really advanced optical methods for causal interrogation of plasticity, to machine learning assisted behavioral screens. So we can do it all to really further develop the top compounds that we're interested in. So in the space of neuroplasticity, it's going to take the work from all of us. I'm just showing a few examples of companies that are really oriented towards the neuropsychiatry applications. And I think here we have a somewhat unique niche with the singular focus on activity-dependent spine remodeling, while maintaining a lot of significant circuit neuroscience context that I'm happy to elaborate on, having excellent speed and scalability, and adaptability across at least a few therapeutic areas. Those most of interest to me include currently neurodevelopmental disorders, neuropsychiatry, which was the original inspiration for creating the screen, as well as potentially some oncology support drug design as well. So the next steps for our company, young as we are primarily connections. We are looking for a CEO. And we're also really excited to build partnerships, particularly those that allow us access to proprietary libraries and new compounds. And we're speaking currently with several startups as well as large pharma companies. I would be glad to raise a pre-seed round right now. I think we're a little early for VC, and we currently have an internal grant pending with Northwestern University. Over the next year, our goals are to further validate this platform, although it already looks really good compared to any NIH-based metrics for high throughput screening. I'd like to expand sensor IP and build proprietary data banks. And the first applications we're considering are seizure disorders that also, I'll be happy to talk about why that is in the Q&A. And with that, thank you so much for your attention.